Hello everyone and welcome to our webinar. In my talk today, I will present you our solutions for myeloid cell research with a focus on macrophages and neutrophils. This is our disclaimer. As these illustrations show us, myeloid cells are diverse and also beautiful. They possess different shapes and sizes, as you see here. But beside that, myeloid cells possess different important functions that I will briefly touch in my next slides. Myeloid cells are present everywhere. So you find in the liver Kupffer cells and dendritic cells. And if you look at the spleen, you find red pulp macrophages, dendritic cells, as well as granulocytes. In the lung, you find alveolar macrophages and also dendritic cells. Present in the blood, you find granulocytes, monocytes, and dendritic cells. And an example of myeloid cells in the bone are osteoclast. Yeah. And in the peritoneum, we have peritoneal macrophages and dendritic cells. And in the skin, we have the famous Langerhans cells and skin macrophages. And in the most important organ of the body, the brain, we have tissue-resident macrophages, specifically named microglia, and also perivascular macrophages. In diseases such as cancer, we have tumor-associated macrophages. These are TAMs, tumor-associated neutrophils, TANs, and myeloid-derived suppressor cells, MDSCs. Yeah, please note these are examples and not restrictions. This widespread presence of myeloid cells in different organs immediately gives us the indication that these cells are very important. So let's have a look together at some examples of myeloid cells functions. Myeloid cells have important functions in health and diseases. For example, in the health state, yeah, so for example, in healthy state, tissue macrophages are highly specialized in removal of dead cells and cellular debris and thus maintain proper organ function. During infection, macrophages engulf pathogens and destroy them, while neutrophils are highly motile and quickly react to chemical signals such as cytokines secreted by other immune cells like macrophages during infection and migrate to the site of infection. Neutrophils have cool methods to attack and kill pathogens, including the formation of neutrophil extracellular traps, as you observe here in this image. And neutrophils can also do phagocytosis to destroy pathogens, or they can release antimicrobial agents from their granules, so to destroy invading pathogens. And in cancer, myeloid cells have positive or negative outcomes. They can contribute to tumor progression by suppressing anti-tumor immune cells. Here I'm referring to myeloid-derived suppressor cells. And research has also shown that macrophages may have cytotoxic activity to kill tumor cells directly. So myeloid cells have also been shown to affect cancer therapy in positive and a negative way. So now let's talk about the sample preparation solutions for myeloid cell research. So careful preparations are definitely important for getting reliable results. So what are the challenges for myeloid cell sample preparation? Time is an important factor. Some researchers process several samples which require long preparation time. Having low number of viable cells is a big challenge in many experiments, and also epitope degradation due to improper sample preparation is a problem for downstream applications, for example, flow analysis or functional analysis, where the cell surface receptors are needed. Having inconsistent yield is a challenge for reproducible result. As a solution for this challenge, I introduce you our gentleMACST technology and associated tools. So the gentleMACST dissociator is a benchtop instrument for the semi-automated dissociation of tissues into single-cell suspensions or thorough homogeneous. Yeah? We have two types of unique gentleMACST tubes used with the instruments, and these enable the time-saving and easy dissociation of homogeneous of tissues in closed system. So the combination of the mechanical disruption using the gentleMACST and the different tubing system with the enzymatic digestion using optimized and highly purified enzymes in this technology enables efficient dissociation. So speed, viability, epitope preservation, and reproducibility are guaranteed. So here I will cover solutions for human and mouse monocytes and macrophages. And I will start with the solutions for the human samples. So here we have a wide range of solutions for samples obtained from different materials, from peripheral blood, whole blood, buffy coat, LRC, leukoplaques, and PBMCs. So solutions for monocytes and macrophages separation shown here in blue, and cell culture solutions for monocyte differentiation into macrophages, solutions for macrophages polarization. This includes cytokines and growth factors shown here in red, and different antibodies for the analysis of these cells shown here in green. Now, let's start with some of the cell separation challenges. Cell sorting requires long time. Yeah. So, and low number of viable cells, which affects various downstream applications, is also a challenge. And a specific cell activation, of course, you don't want to have that on your separated cells for reliable downstream applications. And having few cells of interest after separation is a challenge for conducting your experiments. So now let's talk about our cell separation solution for these challenges. So we offer several solutions for the isolation of monocyte subsets, including the pan-monocytes, variable options of the isolation of classical monocytes and non-classical monocytes. Yeah, I'm not going to go here into details since my colleague Anna mentioned that before for the generation of DCs, but of course we also need monocytes, yeah, for the generation of macrophages. And in this slide, you observe the isolation of monocytes from human PBMCs using CD14 microbeads, which is a positive isolation method. So positive selection means that during separation, monocytes are magnetically labeled, retained within the column, while unlabeled cells flow through. And after a washing step, the column is removed from the magnetic field of the separator, and target cells are eluted from the column, as you observe here. As you observe in the flow analysis result shown here on the right, purity and high recovery of the isolated CD14 positive cells have been obtained using the CD14 microbeads. And now, before I introduce you to our straight from solutions for monocytes, let me say some few words about the straight from technology. The straight from technology enables the separation of highly pure cells directly from different blood sources with all the benefits of MACSxpress microbeads, as well as easy and fast enrichment of leukocyte. So no need for density gradients, no need for cell counting, and no need for washing, and also no erythrocyte lysis is required. So this technology enables multiple separations from one sample. Yeah. So for example, it permits the isolation of CD14 monocytes. Yeah. So you take the negative fraction, and you can from the negative fraction you can isolate CD8. T cells, you take the negative fraction and isolate CD4 T cells. And all this very fast and efficient in just 1 hour time. So CD163 MicroBead Kit human. This kit allows the enrichment of human cells the enrichment of human cells from sample, for example, healthy or tumor tissues based on the expression of the CD163 antigen. So we know that CD163 is most abundantly expressed by mature tissue macrophages and peripheral blood monocytes. You observe here results of the CD163 positive cells isolated from ovarian carcinoma using the CD163 microbeads. And after isolation, the cells were stained with CD163 PE and 206 fit antibodies and analyzed by flow cytometry. And the isolated cells are ready to use for downstream applications. Now, let's move to the solutions for mouse monocytes and macrophages. Here we also offer several options for different sample sources, from cell separation to cell culture to cell analysis. And here are the solutions for mouse monocytes and macrophages in the lung. Yeah, cell separation, cell analysis solutions. Now, the anti-F480 MicroBeads UltraPure, which is a very good option for the isolation of mouse macrophages from a spleen or tumor tissue. And here below is flow data showing the separation of F480 positive cells from the spleen, and from a tumor tissue. This result was a collaboration work with Dr. Subra Biswas in Singapore. This is just an example of a publication showing that macrophages isolated by anti-F480 MicroBeads UltraPure are ideal for downstream molecular analysis. Here, macrophages isolated from tumor, from breast cancer xenograft using the F480 macrobeads were analyzed for innate immunity pathways using the NanoString platform. So the general message of this result is that the treatment with different drugs modulated macrophages gene signature. And for the isolation of mouse monocytes and macrophages from blood or lymphoid tissues, we have the CD11b MicroBeads UltraPure. You observe here in the dot plots CD11b positive cells isolated from a spleen using CD11b MicroBeads UltraPure mouse is stained with CD11b Fitch and anti-GR1P antibodies and analyzed by flow cytometry. In the first dot plot, the cells, you see the cells before separation. In the second dot plot, it shows the flow through without the CD11b positive cells that are returned in the column. And in the third dot plot shows the isolated CD11b positive cells. So to wrap up the section of monocytes and macrophages, our solution for these cells guarantee time saving, optimal viability and functionality, as well as reproducibility. Now, I would like to talk to you about our solutions for neutrophils, and I will start with our solutions for the human samples. So we provide several solutions for human neutrophils research. And here you observe our cell separation solutions written in blue and our cell analysis solutions written in green. The challenges I mentioned in my previous slides for monocytes and macrophages exist here as well, so I don't need to go through it in details again. So what are the solutions for these challenges in case of human neutrophils? Well, let's have a look together at our MACSxpress technology. Human neutrophils are usually obtained by density gradient centrifugation. However, this method is exceedingly time consuming and frequently results in neutrophils populations that are contaminated by other leukocytes. Yeah, these are mainly eosinophils. And with the MACSxpress technology, untouched human neutrophils can be isolated from the whole blood within 20 minutes. And in this method, so the erythrocytes are aggregated and sedimented, and non-target cells are returned at the tube wall by immunomagnetic depletion with the MACSxpress beads and the help of the MACSxpress separator. And then the untouched highly pure target cells can easily be collected as you observe here in the illustration. Now, in these results, you see that human neutrophils are efficiently isolated from human blood using MACSxpress neutrophil isolation kit. In the upper dot plot, you observe that cells before separation, and in the lower dot plot, you observe the cells after separations. And here, after separation, the cells were stained with CD15 and CD16 antibodies, which are markers for human neutrophils. The kit allow isolation of neutrophils with negligible number of monocytes and reduced number of eosinophils, yeah, as compared to the density gradients with high purity, recovery, and viability. And on the right, you see all the nice features listed, yeah, using this kit. Here are the functionality of the neutrophils isolated with the MACSxpress technology was tested. The neutrophils isolated with the MACSxpress technology show high ROS production upon stimulation with E. coli, red bar, and with PMA. Stimulant, this is the light bar as compared to the density gradient. And these data were obtained from flow cytometry analysis. Now, let's go to the solutions for mouse neutrophils. Here we also have rich solutions for different tissues. And here I introduce you the anti-LY6G MicroBeads UltraPure for the isolation of mouse neutrophils from lung tissue, blood, or bone marrow. And in the dot plots, you observe that cells before separation and the effectively isolated neutrophils indicated by CD11b and anti-LYG6 staining. Now, these are results from a very recent publication where they conducted transcription analysis of neutrophils isolated from mouse liver and blood. So, and the graph on the right shows gene expression analysis of mouse neutrophils, and the dot plots are representative flow cytometry profiles of isolated neutrophils from blood or from the liver. The message here, the neutrophil isolation, the anti-LYG6 MicroBeads UltraPure mouse are ideal for transcription analysis. So I'm getting close to the end of my presentation, and in my last slides, I will talk about our cell analysis solutions for myeloid cells. So once you get your cells of interest, of course, you want to analyze them. And here in the cell analysis part, we also have several challenges. One of the challenges could be that the antibodies are not highly specific, so they are not effective in capturing the antigen of interest. Moreover, having high background signal where antibodies detect signals that are not specific for the antigen, And one of the reasons of high background signals is non-specific binding of antibodies to the Fc receptors found in myeloid cells such as macrophages, monocytes, and dendritic cells. And this is actually one of the reasons that make these cells, difficult to analyze, due to their similarities. So the solution is our REAfinity antibodies. These are recombinantly engineered antibodies with guaranteed lot-to-lot consistency. They are highly pure. They consist of only one universal isotype control. And most importantly, these antibodies are engineered to lack Fc part, which leads to the preservation of unspecific binding. This is an example of human macrophages phenotype using the REAfinity antibodies. And the results show that CD163, CD209, and CD200R are highly expressed in M2 macrophages. And CD68 is highly expressed in M1 macrophage. So, and the table on the right shows panel suggestion for macrophage phenotyping. At this point, it's appropriate to mention that we have a complete workflow suitable for macrophage assays, for macrophage generation to differentiation and phenotyping and analysis. This workflow is available as an application note, which you can find online on our website. The link to the app note is also included here. You can just check it when you receive the PDF version of this presentation. Now, I will wrap up my talk, and here are some take-home messages. Please remember our gentleMACST dissociators and the combined dissociation kits for optimal myeloid cell sample preparation. Sample preparation definitely matters. And we also, our optimized separation solutions, these are positive isolation, untouched isolation, as well as the straight from solutions, which are suitable for several downstream analysis, omics, cell culture, flow phenotyping, etc. and etc. And also our REAfinity antibodies that guarantee consistency, standardization, and high specific signals. And most importantly, our experts are happy to guide you through the entire workflow for your myeloid cell research. Here I included a list of useful materials for myeloid cells that can benefit you. And for deeper and more extensive information, please visit our website or contact your local sales organization. So thank you very much for your attention.